I'd like to welcome everyone to this week's Extracellular Vesicle Club. So this week's EV Club is featuring Carmen Campos Mar Valés-Gómez, and they're going to be presenting some work that they've done recently on liquid biopsy. So how can we take a very small sample of a biological fluid and profile the extracellular vesicles in it? So Carmen, thank you so much for being willing to present your work to us. I'm going to allow you to start the screen sharing now. And I would just like to ask everybody on the call today to put your comments and your questions for Carmen and Mar into the chat box. And then at the end of the presentation, we will allow unmuting so that you can interact with our presenters today. And we'll look forward to some very stimulating discussion. Carmen, please go ahead.
Okay, thank you. Hello, everybody. My name is Carmen Campos Silva. I am a PhD student at Dr. Mar Valés-Gómez lab in Madrid. And today I'm going to talk about simple immunoassays for EV liquid biopsy in microliters of plasma and other body fluids. So I will be presenting mainly this article that we published this year, but also I would present some of the previous work that led us to perform these studies, and also some of the latest work that we have been doing that is not published yet. But first, let me tell you why we started all these projects, because in our lab, the main topic we are focusing on studying a group of immune modulatory proteins that are expressed by tumor cells, and they can be released by metalloproteases as soluble proteins. And we have also described that they can be released recruited in extracellular vesicles, and also they can modulate the immune system, cytotoxic lymphocytes, in these forms. So we thought it was interesting to study these forms of these proteins in patient samples, in cancer patient groups. But we found that the methods to perform this type of studies had a number of limitations. So that's when we started these projects that I'm going to talk today. So as you all know, EVs are released to body fluids, to different body fluids, and in them you can find a number of molecules which can provide interesting or important information for the clinics. And this is what is called liquid biopsy. So liquid biopsy in the case of cancer can provide important information for different clinical features, such as diagnosis, prognosis, the selection of personalized therapy, or monitoring the disease, or even in some cases the immune competence of patients, as I was commenting before. Much easier than conventional invasive biopsies. So a lot of biomarkers have been proposed in EVs, and the problem is just a few of them have been validated, and we really need validation studies. And this is hampered by the lack of high throughput techniques to be translated to the clinics. So as you know, in order to study EVs, normally you perform an EV concentration or isolation, and the method to use depends mainly on the aim, because the different methods can yield different specificity and recovery. And so not only that, but also some alterations of the samples of the EV subpopulations can occur during these processings. So we could be enriching some EV populations and discarding others. Also, the EV-associated molecules could be altered. And I borrowed this slide from Dr. Kenneth Witwer from the past week workshop on blood EVs, because I loved it. Because, you know, when we are studying our EV pellets, biomarkers in them, sometimes we don't really know if they are actually inside the vesicles, if they are on the vesicles, or if they are loosely associated with the vesicles. So some of the biomarkers we are studying may be at the EV corona, and with this processing, we could also be altering those. So last but not least, all these methods require specialized personnel in most cases, or specialized machinery, or either larger starting volumes, which is not feasible to translate to the clinics. So this is a survey performed by the ICEF, rigor and standardization committees to the people of the ICEF society. And yeah, these are the methods used by most people. So for EV characterization, and they include Western blot, single particle tracking, protein concentration measurement, electron microscopy, and others less used. And here I show some examples from our publications on EV characterization by those methods. But yeah, the problem is that most of these characterization methods require a previous EV enrichment. And again, we have the same problems that I described previously on EV concentration and isolation. So talking about EV characterization methods, other options are immunoassays, and a number of immunoassays have been described for EV characterization. Here I show some examples, such as ELISA. And the thing is that in this publication that is also from our laboratory, in which we described lateral flow immunoassays and ELISAs for EV characterization, in this during this optimization, we noticed that EVs are much more complex than soluble proteins, and because most of these assays have been developed for the detection of soluble proteins, we have to take into account a number of considerations, since in the vesicles we have different different proteins and different amounts of each protein. So we observed that competition events could occur between the antibodies, and also steric impediments occur. So yeah, and another observation in some of the described methods for bead-assisted immunoassays, in this case flow cytometry, some approaches use different beads against different proteins to capture different proteins. And the thing is, for example, an anti-CD9 capture bead will capture a lot of EVs that could also have other less abundant molecules, such as tumor-associated markers. And then those bound EVs are going to be no longer available for other beads that capture less abundant proteins. So we have to take into account, yeah, that vesicles are more complex, and this kind of competition events and all this can happen. And also, most of the described assays require, again, a previous EV enrichment, and this highlights that sensitivity issues are going on with these approaches, because the advantage of immunoassays is that in theory you don't need to isolate the EV fraction, because you can select it specifically from a complex sample with different molecules. So all of that led us to start this project, in which the main objective was the optimization of high throughput sensitive immunoassays, and we tried to study the key factors affecting the sensitivity of these immunoassays for EV cancer-associated protein analysis directly in biological samples that don't require a previous processing. And this led us to study the physicochemical properties of EVs in solution and its relevance for EV immunocapture. And in the latest months, we have also performed some comparisons of these bulk EV immunoassays that we have previously optimized with technologies that have entered the field recently, such as ExoView. So as I said, we started optimizing these immunoassays. We started with ELISAs, and we noticed that yeah, for example, the different combinations of capturing and detector antibodies is an important factor for the sensitivity, and other key factors that I will talk a little bit later. And also we moved to flow cytometry, because it's another technique widely used in the clinics. And I also borrowed this slide from last week workshop on blood EVs from Edwin van der Poel, whose talk was on approaching clinical feasibility with EV flow cytometry. And he mentioned that specialized instruments that are developed for the detection of extracellular vesicles, so they have a low flow speed and all this, they have a limit of detection of around 50 nanometers, and other modern conventional, modern clinical cytometers have this limit of detection of around 100 nanometers. So most small EVs fall below this limit of detection. So still we need complementary methods to study these, to study biomarkers in small EVs. So here I love this picture because it depicts an average size of a cell of around 6 micrometers, together with the average size of a small EV of around 150 nanometers. And if you imagine that flow cytometers were developed to detect this type of particles, you see how difficult it is to detect these small particles. And also, not only the size, but also the fluorescence, because you can imagine the amount of antibodies binding here are much less than the ones binding this other type of particle. So we started a collaboration with a company to optimize a bead-assisted flow cytometry method for the detection of proteins in EVs. And the rationale was very similar to the ELISA, but now we had 6 micrometer latex microspheres that were also magnetic and were coated by a capturing antibody against, for example, the protein of interest. And then we used detector fluorescent antibodies against tetraspanins. So because a picture is worth 1,000 words, here you see one of these 6 micrometer beads with these dark spots are iron nanoparticles to make them magnetic. And then you see these small gray round shaped things that are binding, and those are the EVs binding to the antibodies coated on the bead. So we perform titrations of different EVs derived from different cell lines. In this case, it's a prostate cancer cell line, and we confirm that this was a semi-quantitative approach. So also, this is all published in this paper in 2019. We also determined some of the key factors for a sensitive detection by these immunoassays. So we observed during the incubation for capture of the EVs, factors such as time and temperature were really important. Here you see the difference of the detection after an overnight incubation of the beads with the EVs, or 2 hours, and also 4 degrees and room temperature have different detection. Not so much agitation. In this case, it was horizontal agitation. And also about bead number, this is something really interesting, because most studies optimizing immunoassays, bead-assisted immunoassays, they use a number of beads, and they don't even say why they use this number of beads. So one can think that the higher the number of beads that you put, the better, because you will capture more EVs. But in fact, for flow cytometry, this is not the case. It's actually the opposite, because if you put more beads, then less EVs will bind per bead. So you will have a lower signal. And in fact, here you see an example in which we used 6,000 or 12,000 beads with the same number of EVs, and you see with more beads, you have a lower signal. So in this paper, we describe also some theoretical calculations on the number of beads that we should use, also based on the diameter of the beads that we used, and so. And also, we show that antibodies combination is important, as I said previously. For example, here we were trying to detect EPCAM, and we found that capturing by this is a tumor-associated protein, and capturing by this less abundant protein and detecting by the most abundant proteins, such as tetraspanins, rendered a better signal with less amounts of EVs than the opposite combinations. Also, we demonstrated the specificity of the captured beads. In this case, for example, we used EVs that were negative for EPCAM, and you can see that the signal of the EPCAM capturing beads is very similar to the isotype beads. So we then moved to biological body fluids. Because this was the aim in the end of these studies. And in this experiment, we use urine from prostate cancer and benign prostate hyperplasia patients. And we performed parallel experiments in which we took 500 microliters of those urines and used them for the bead-assisted immunoassay and flow cytometry. We use beads capturing EPCAM or isotype beads and detection with anti-CD9 antibodies. And then those relative fluorescence intensities, you see here a heat map with those intensities. And we also took 8 mL of those same samples and performed an EV isolation by ultracentrifugation and Western characterization. And there we saw that the signals we obtained for EPCAM correlated with the signals obtained by the immunoassays. And also in some cases, we could observe a signal in the immunoassays, whereas in the Western, we couldn't see that signal after the EV isolation by ultracentrifugation. So like this, we validated this method. We demonstrated it's faster, it's easier, you use much less volume, and you have a better sensitivity. And also the most important thing that was our aim is that this method has a high throughput potential, so to be translated to perform validation studies. So now moving to the article I'm presenting today from this year. This study started with the observation that not all the EVs that we were providing during the overnight capture were captured by the beads. And we demonstrate that with this experiment in which we incubate the EVs with the beads for an overnight as usually. And the next day with a magnet, we recover the magnetic beads with the bound EVs and we stain them for flow cytometry. But instead of discarding the supernatant, this time we recovered it and incubated it with new fresh anti-CD63 beads. So the same as anti-CD63 beads used before, but new beads. And again, we incubated that to see if there were still the EVs binding to those beads. And again, the next day, we recovered the beads with the bound EVs and stained that for flow cytometry, and we repeated this process for several days. And so we observed that each next day, we could still detect EVs binding to the new fresh beads. So our hypothesis was that there could be a lack of interaction of the beads with the EVs, since the beads are 6 micrometers and they fall to the bottom of the tube relatively fast, and the EVs probably stay in suspension. They stay in suspension. So we started thinking about possibilities to enhance this interaction, and because horizontal agitation did not seem to help a lot to increase the signal, we started thinking about the EVs as colloidal suspension particles in which they are stable. And so we decided to try chemical molecules that have been used to destabilize colloidal suspensions to use them in these EV suspensions to see what happened there. So we use Polymer and polyl-lysine that are cationic polymers used in colloidal, in the field of colloids. We first characterized the zeta potential of the EV suspensions when we added these cationic polymers. And so we observed that there was an increase towards a positive value in the zeta potential of the EV suspension. And this was expected, but we also found, and this was interesting, that there was an increase in the average diameter of the EVs when we added these cationic polymers, as measured by dynamic light scattering. So we investigated that by electron microscopy, and we saw that indeed, when we added these cationic polymers, we were inducing clusters of EVs that were being formed there. And also we analyzed by analytical ultracentrifugation the sedimentation coefficient of the EV suspensions when we added these cationic polymers, and we observed an increase in the sedimentation coefficient. So with these polymers, we are destabilizing the EV suspensions, and they are falling at a higher speed. And because these ion interactions have been described to be reversible, then when we perform the different washes during the protocol, these ionic interactions could be reverted. So we tried to use those cationic polymers in our bead-assisted immunoassays. Here you have an example capturing by EPCAM and detecting CD81 in EPCAM positive EVs. And we saw that when we added polybren at 8 micrograms ml, we had a significant increase in the signal with respect to the untreated samples. So indeed, this could help in the detection of the EVs. And this finding also led us to think about different approaches to try to enhance the interaction of the EVs with the beads. So we tried different assay geometries, so different platforms, and compared conventional cytometry tubes with V-shaped plates, U-shaped plates, and flat bottom plates. So also we tried with different final volumes of the assay. And indeed, we found that flat plates render the best signals. And also we could reduce the volume, the sample volume, down to 20 microliters, and we still had a much better detection than the one with a 100 microliters sample in a conventional flow cytometry tube. So with those settings, we moved again to body fluids, and these experiments, previous experiments, were performed with EVs isolated by ultracentrifugation. And now we used just 12 microliters of healthy plasma, and we wanted to determine the limit of detection for EPCAM, this tumor-associated protein. So we used lung cancer cell line derived EVs that we know are positive for EPCAM by Western, and we added it at different concentrations as measured by nanosite to this healthy donor plasma. And we determined that around 3 × 10⁶ EVs per microliter were enough to see a positive signal for EPCAM in these assays, which was indeed a good limit of detection. Also, we tried adding cationic polymers to these assays with these settings, and we saw that we could still increase, not as significant, but still significantly could increase the detection in these settings in which the interaction between the EVs in the sample and the beads was already increased. So we moved to real samples from lung cancer patients. And we had 3 groups of donors. In this case, we have healthy donors, initial stage lung cancer patients, and advanced stage lung cancer patients. And in just 12 microliters of plasma, we could see CD63, CD81 positive EVs. And also we observed that advanced lung cancer patients had higher levels of those than the other 2 groups. We also detected EPCAM CD81 positive EVs in the plasmas from some of the initial lung cancer patients, and also MICA positive CD81 positive EVs. MICA is one of these tumor-associated proteins that are immune modulatory that we study in our laboratory too. So with this, we validated the method so that it has a high throughput potential to perform validation studies. But we tried to perform an EV isolation from these plasmas and detect by Western the signals for EPCAM and MICA especially. But by Western, we couldn't see a signal for any of the patients. For those proteins. So we moved to other cancers that have been described to express larger amounts of EPCAM, such as ovarian and breast cancer. And we obtained plasma from those patients and again perform a similar experiment. So we use just 12 microliters for the immunoassays, or 200 microliters of plasma for an EV isolation by ultracentrifugation. And so we could see that, I have to say here, this time with these pellets, we analyzed them both by Western and by bead-assisted flow cytometry to have the results for exactly the same sample. So we saw that the correlation of the signals on the immunoassays was good with the signals from the Western. But again, we couldn't see a signal for EPCAM even after long exposure, only for the positive control that we added. These are EVs derived from this lung cancer cell line that we know they express EPCAM. And though we detected a signal by the immunoassays, we couldn't detect a signal by Western. So we moved again to other type of samples that have been described to have large amounts of vesicles, and specifically EPCAM positive vesicles, and those are ascites from ovarian cancer patients. And again, we performed the same type of experiment, and finally we could see a signal for EPCAM by Western, and we saw that it correlated with the signals obtained in those same pellets by bead-assisted flow cytometry. But yeah, here we also observe that the signals obtained directly in the non-processed sample were somewhat different to the signals obtained after the isolation by ultracentrifugation. So even though the signals obtained by both methods in the pellets correlated, the signals compared to the non-processed samples were to some extent different. So again, this highlights that when you perform these isolation methods, you can be altering the sample and selecting certain EV subpopulations. So like this, again, we validated this method for ascites and plasmas from cancer patients. And also in the latest months, we have performed these studies to compare these optimized immunoassays with ExoView technology. For this, I got a grant from the Spanish Society on Extracellular Vesicles, a mobility grant, to go to the lab of Dr. Maria Pardo to perform these studies with ExoView. So briefly, ExoView use microchips that are coated with anti-CD81, CD63, CD9, or isotype controls. And you can also coat some spots with your antibodies against your protein of interest. In my case, it was MICA and ULBP2. These are these tumor-associated proteins that are also immune modulatory proteins that we study in our group. And yeah, so you can also coat with those. And then you go with fluorescent antibodies against tetraspanins or your protein of interest. And so the method is very similar to the immunoassays that we optimized, but then you go with a high resolution microscopy and you can see the spots from the fluorophores. And also you can obtain, in a different channel, you can obtain an interferometry measurement that is marker independent measurement. So for this, we isolated the EVs to perform these studies by ultracentrifugation, and then we characterize them. In this case, I used metastatic melanoma cell line derived EVs that are positive for MICA, and also these lung cancer cell line derived EVs that have a negative signal for MICA by Western. So I use these ones as a negative control. So yeah, we studied the limit of detection by ELISAs for this protein, MICA, in the EVs, and we found that around 5 by 10 to the 6 EVs per microliter could be detected either in PBS casein or in diluting the EVs in serum, in healthy donor serum. And so here you see that increasing amounts of the negative control EVs rendered a lower signal than the one obtained with these amounts of EVs from the metastatic melanoma cell line that are positive for MICA. So finally, we studied these EV preparations, the same EV preparations by ExoView, and we could see that capturing by MICA, this protein of interest, and detecting by tetraspanins, we could see a positive signal compared to the isotype. And also the signal was higher than in the negative control EVs that don't express MICA, or at least we cannot detect that by Western. And so we saw this higher signal. We confirmed that these EVs express MICA and also could perform colocalization studies and studies on the size of these particles. Yeah, but something here that was also interesting to see was that when we tried to detect by an anti-MICA fluorescent antibody, the signal was very low. It was even comparable to the IgG. And this again highlights the importance of the combinations of antibodies. So it's better when you capture by the scarce protein and then you detect by more abundant proteins in order to have a good detection by these immunoassays. And so we also try to determine the limit of detection for this protein by ExoView. And we found that when we used around 2 by 10 to the 5 EVs per microliter as measured by nanosite, we saw a signal that was pretty similar to the isotype. So we may be near the limit of detection. And so we can say that ExoView is 10 times more sensitive than these bulk EV immunoassays that we have previously optimized. And it also allows an EV marker independent detection, but it's yet not adapted for high throughput screening because of the long times, the long reading times, and also because of the costs of this technology. So yeah, so as conclusions, I can say that the strengths of this work is that we have described critical parameters for immunoassay sensitivity, such as antibody combinations, the numbers of beads, the capture incubation time and temperature, and also the geometry of the assays. We also have detected cancer-associated markers and tetraspanins, of course, directly in minimal volumes of non-processed body fluids. And also, these methods have a high throughput potential in order to perform validation studies. So the opportunities of this work is that it has an easy translation to the clinics and for validation studies. So we really invite people that has an easy access to patient samples that to try and yeah, optimize these methods, taking into account all these considerations, because you can really increase the sensitivity and yeah, and to perform these validation studies, because we really need that. And also the colloidal behavior of the AV suspensions can be exploited for immunocapture, as we demonstrated, but probably for other different applications, the use of cationic polymers is also interesting. And the other thing is that cationic polymers may be useful for qualitative studies, but in the case of semi-quantitative studies, probably we have to consider different optimizations for different molecules, for different markers in different body fluids. And finally, we can say that ExoView and probably other single vesicle resolution technologies are more sensitive than bulk EV immunoassays. and allow EV-independent marker detection, but they are not yet adapted for these high-throughput screenings. So still, these are complementary techniques, and probably for validation studies, these methods are going to be helpful. So with that, I want to thank the people from my group, my supervisor, Dr. Mar Valés-Gómez, and all our collaborators, and of course you for your attention. And here you have some of the literature that I have mentioned during the talk. You can check here. We describe thoroughly our protocols, and also you can contact us to talk about anything or for collaborations. We're happy to talk. So thank you very much.
Thank you so much, Carmen, for the presentation. And maybe I can start out by just asking, Carmen, where do you think the future is taking us in terms of multiplexing? So in terms of how many different markers we should be looking at on EVs, what do you think is the ideal there? Because, we have to strike a balance between the sensitivity and the information that we're getting out of the liquid biopsy sample.
Yeah, that's an interesting point. What we have noticed is that that with EVs we have to be careful and take into account different considerations, because, for example, with the multiplex assays that use beads against different capturing different proteins, we can induce competition events, so that the EVs expressing a scarce marker can be captured by an anti-CD9 capture bead, and then this EV is no longer available to bind to the bead that is capturing this scarce protein. So we have to take into account that when trying to optimize multiplex technologies, and also in terms of antibody detection. Also, if you go with a bunch of antibodies, incubating them all at the same time, you can have again competition events, and also even aesthetic problems, aesthetic impediments. We have observed that in our publication in 2018, we have described this kind of aesthetic issues in lateral flow immunoassays. And so we have to be careful with multiplexing, because we have to keep in mind that we have a complex molecule that is not a soluble protein. It's much more complex. So yeah, we have to be careful with the multiplexing methods.
I can add something, Carmen. What I would say is also in different immunocapture experiments, you have different order of incubation. And for example, in lateral flow, you incubate first with the capture with one antibody, and the full vesicle can be surrounded by the antibody, and then you make it flow through the membrane. So in this case, it's very important to think about the competition, because if you have a very abundant epitope covered by your antibody, you are not going to be able to stop on the membrane with the other antibody. So it is not the same ELISA, lateral flow, or any other method. Like, for example, in ExoView, you have the same problem, and we have now some experiments in mind with single antibodies compared to multiple antibodies, because it could happen that we are not detecting so much MICA because maybe we have the problem of competition.
Yeah, well, thanks for the insights there to, I think, this very important question, important set of considerations. So we have some questions that are starting to come in here in the chat box. I'd first like to go to James Drummond. James, could you please ask your question? And actually, I need to, let's see if we can risk letting people unmute. hopefully no one else is here. Okay, so James, go ahead and unmute now if you can.
Okay, I don't know why my video, at least it's not working on my screen, but I just was wondering about the polymers you're using. You used all the cationic ones, polylysine, polybrine. Is it necessary to use a charged polymer, or can you get away with other non-charged ones?
Okay, we haven't tried. The ones we have tried are the cationic polymers, and the advantage of this is that these ionic interactions can be reverted by washes or are theoretically reverted if you decrease a lot the ion concentration and the pH and other parameters. So the good thing about using these ionic polymers, cationic or polymers, is that you can tune these interactions, and this is the advantage compared to other polymers such as polyethylene glycol, that they have water excluding properties, and what they do is aggregate everything, and then it can be complicated to disaggregate these the vesicles or the different components of the biofluid. So that's the advantage of the charged polymers. But we haven't tried with we tried with polyethylene glycol, but we didn't have a good outcome because the signals were much dirty. The isotype controls signals increased, and we didn't went further with non-charged.
Okay, thank you very much.
Thanks for the question, James. And now a question from Anthula Lazares.
Hi, Carmen. Thank you very much for a very interesting talk. And also, you covered a lot of information and you really articulated it very well. So I really appreciate that because it can be very complex. I'm interested in intravesicular proteins. Are you looking at staining for something intravesicularly, I should say, intracellularly, especially if we're looking for like biomarkers for different cancers, diagnostics and all that? And if you have or you haven't, what kind of challenges would you foresee?
We have, this is really interesting. We have had this in mind for a long time now. We have not yet performed permeabilization assays to detect intravesicular proteins, but we have this in mind, and actually in theory it's perfectly possible. For example, in ExoView they also detect intravesicular proteins with this kind of antibody combinations and so, and a permeabilization more or less conventional as the ones with cells should work for this kind of assays detecting intravesicular proteins too. But we have not data yet on that.
Okay, and just a comment for the EBCAM, we're finding some of our tumor cells, as they start to secrete vesicles, they differentiate, so they lose expression of EBCAM. So I'm actually staying away from EBCAM for some of my cancers, because I find that when we stain, for example, human tissue, we often see variations in EBCAM levels. So it's just a comment, and if you've had that experience as well.
No, it's a relevant point. Actually, in the essays in which I don't know if I can share that, in which we tested EPCAM in lung cancer patients, we observed that only in the initial stage, so the first stages lung cancer patients, we found EPCAM positive EVs. And it's tempting to hypothesize that this could be due to an epithelial to mesenchymal transition in these patients, that they have also metastasis. But because this is a very, very low number of patients, we cannot conclude anything from this data, but that's an important point. Yeah, of course, we started with EPCAM because it's a widely used tumor-associated protein, and we had these lines that express EPCAM. But indeed, about the relevance as a biomarker, I cannot say anything about it.
Thank you.
Thank you.
All right, we have a question here from Praveen, who has background noise, and so I'm going to ask the question. And so Praveen says it was a nice presentation. Thank you. What was the yield you observed when EVs were isolated by ultracentrifugation from microliters of plasma or urine? Is that something that you looked at, the yield?
Well, actually, we performed this ultracentrifugation on 200 microliters of plasma. The 12 microliter assays were the immunoassays, but for the ultracentrifugation, we used larger amounts, around 200 microliters. And then for Western, we used, I think it was 5 microliters from this EV preparation. We resuspended this EV preparation in 15 microliters and then used 5 from those for Western. And then for the immunoassays, I don't remember right now, but I think we used less than 5 microliters, like 2.5 microliters or so, but less than for Western. So I cannot say much about the yield. Actually, we use ultracentrifugation because most of our studies then use immunocapture or immunoassays. So then we go, we specifically see what we want to see. But yeah, I cannot talk much about the yields, but this is what I can tell.
Good. Okay, so these Western blots were basically coming from about 67, whatever microliters of original.
200 microliters. Yeah. 200.
Oh yeah, sorry, and then you were using about a third. Yeah, so that makes sense. All right, so thanks for that. Let's see, Tanina, you have a question.
I do. Very nice talk. I really enjoyed it, but I have to rewatch it just to get the full benefits of it. I was just wondering if the method that you established, well, actually, the first question is EPCAM on the surfaces of EVs abundant as tetraspanin, or is that a rare event marker?
It depends on the cell line. For example, on PC3 derived EVs, it's a very scarce marker, but scarcer than tetraspanins. But in the case of these H3122 lung cancer cell line derived EVs, it was almost as abundant as CD63. So it really depends on the cell line. Yeah.
I see. Have you been able to test this method with any marker that is like between 1 and 10% of incidence? Because sometimes we're looking for this needle, that is not as abundant as tetraspanins. And I was just wondering if your method will work for this kind of markers.
Sorry, I didn't get the last part of the question. So can you repeat it, please?
Yeah, no problem. I was saying that I was just wondering if you have tested the method with a less abundant marker and see if the sensitivity is still there and you will be able to detect it.
Yeah, sure. With MICA is a really scarce protein in comparison with the tetraspanins. Actually, you can see here, for example, in these ELISAs, we diluted the EVs in PBS casein, and you see. Well, for example, when we use 1 by 10 to the 6, not 1 by 10 to the 7 EVs per microliter. This is the signal you have for MICA, that is around 0.0 something. And for CD63, CD81, you see the signal is 0.25. So the abundance of MICA it's much scarce than tetraspanins, and you can see it clearly with the immunoassays. Also with I think I have another slide on MICA. Just one second. Here you see, for example, these are metastatic melanoma derived EVs, and this is the abundance of MICA detecting with CD81, and then this is CD63 detecting with CD81. So you see the abundance of MICA is really low, and this is the lung cancer cell line derived EVs and the expression of EPCAM in comparison with CD63. You see that they are similar, but in the case of MICA is much scarce. Actually, you have to put a lot of EVs. You see in the detection with CD63, you see you are putting a lot of EVs to see some MICA. So yeah, we can say we have done these experiments with a very scarce tumor-associated protein in EVs. So yeah, we have these limits of detection with also with MICA.
Thank you so much for sharing that. This is actually very encouraging to go towards the single EV profiling, even for low markers. So that's exciting. Thank you.
Thank you.
Thanks for the questions, Tanina. And with that, I think we can conclude for today. So thank you so much, Carmen and Mar, for this excellent work and for sharing it with us so eloquently. And we look forward to seeing the continuation of this as you proceed. And I will look forward to seeing many of you again in future EV clubs. Again, we will be going to a weekly login now, not using the same one. So please burn this login. We will never use it again, but we will we'll make sure that we continue the resource for the community. All right. Well, thank you, everybody. Have a good rest of the week and good luck with your research. Bye now.
Thank you.